Hello, dear students. In last lecture, we have discussed the funnel device for the solid impingement device. Now, in this lecture, we'll discuss about the sieve type of sampler. So these are nothing but the small hole made on the particular object, so that only the viable count we can get. Or here, perforated plate we can use for the uh, sampling. So here, this type of air sampler in which the petri dish which contain agar medium is placed in a metal container having large number of holes and each having a diameter of about 0.5 mm. So here we have to count the microbial count load in that particular air, so that we have to get the microorganisms which are present in the air. So we have to provide nutrition medium for the growth of organism. So as like now panel device, we have to also provide nutrient agar plate, and above the nutrient agar plate, we have to place a metal container, and upper surface of that metal container, we have to prepare a small hole of diameter 0.5 mm. Air which is introduced into the metal container through the holes, and the microorganisms which are present in the air get impregnate on the surface of the agar medium. The hole present in the container acts as a sieve, not allowing dust and other particles to pass. The petri dish is then incubated for 30, at 37 degrees Celsius for 24 hours at incubator, and after incubation we have to observe for the viable growth of microorganisms. So what happens? This type of air sampler in which the microbial air sampling device which draw the air through a perforated plate. So that is nothing but the hole which we have prepared on the surface of that metal container, which allows or having diameter of 0.5 mm, and which allow only the microorganisms to enter into the container. The airborne particle directly onto the agar surface for culture and counting. Now, it is nothing but the most widely used method for the uh, counting of viable organisms present in the air. How it works? Uh, the air which is pulled through a metal plate containing hundreds or tiny hundreds of small organisms which are suspended in the air, we have to grow on the surface of the nutrient agar plate. Air jet accelerate through the holes. Microorganism impact and agar plate position just below the sieve, so that as the air enter into the container, the microorganisms which are present in the air get trapped on the surface of the agar plate, and as there is nutritional medium, they start forming their colonies on the surface of the agar plate. After that process, we have to incubate the agar plate for the observation of viable visible growth of microorganisms. Colonies are counted CFU, that is colony forming unit, per meter cubic on the basis of that diameter of that particular plate. And this method is also called as single stage impactor sampling. So only one sieve sampler is placed on the surface of that metal container, so that that container is also called as single stage impactor sampler. Now, in the diagram, you can see this is nothing but the simple diagram of the sieve sampler. Only the one single stage sampler we have used for the inauguration of microbes. Apart from that, different methods we can also use, where multiple sampler are used or multiple stage are used, so that multiple stage sampler we can also call this. Now we'll discuss what are the advantages of this sieve sampler technique. It is simple in operation. Direct viable count of microorganisms can be observed after 24-hour incubation. It is already a standardized method, and it is based for the validation of the cleanliness of the particular room. So it is a simple method in operation. So no further glasswares or equipment are needed, and uh, we can observe after 24 hour result for the cleanliness of that particular or validation of a particular room. What are the limitation of this room? Only it can detect the viable organism which are present in the air. Overloading can occur in dirty environment, as there is no filter, directly air can enter into the metal box to that sieve, so that if the microbial load is present more in that particular environment, so we can get the multiple colonies on the nutrient agar plate, so that we can unable to count the multiple colonies which are present on the surface. Particle bounce possibly as directly enter into the it, and not ideal for very high dust areas, as it is nothing but the basic limitation of that particular tube. So it is all about sieve type of method of solid impingement device.